Hello again. We are at the DP stage. We are adding excipient to the DS to make it more easily consumable for use and better effect of the drug on patients. We already have DS from last process. As you see on your screen, we have the DS from last process, and in the DP process, we are going to fill drug in syringes. This is the journey we are going to walk you through with the help of this BPR of DP. With step-by-step instructions, you will learn the process of taking DS and other applicable materials, sterilizing them in autoclave, doing formulation in formulation room, and finally, filling the drug product DP in the syringes. Now, I would like to tell you this DS will be turned into injections. Similarly, you could be working in any company that may produce vials, ampoules, tablets, capsule, syrups, and many more. It is very important to learn GMP principles and core foundational principles of manufacturing, keeping FDA guidelines in mind for patient and drug safety. Now you're getting familiar with the first page of the BPR. As you can see on the screen, this is the first page of the BPR. You have seen this page in DS training as well. Again, this is the most important part of BPR that you need to understand. You will confirm all these entries on the page that are numbered 1 to 10, highlighted in yellow on your screen before you start working on manufacturing operations. Number 1 is the product name. It must be spelled accurately. Number 2 is the product stage. Number 3 is the batch number, and number 4 is the work order number, which you have already learned in DS stage. Number 5 is the batch size or theoretical yield, which is decided before starting the formulation process, based on the material issued. Number 6 is the name of the company who manufactured the drug with the date of the manufacturing. The date when the drug is filled in syringes is the day of the manufacturing. Number 7, similarly, the science team has established the fact that the drug will be stable for a certain period of time. Let's say in this case, if your BPR instructions are telling you that expiration period is 24 months. In this case, you can add 24 months to the manufacturing date and fill the expiration date after the manufacturing activities are completely done. Number 8 is for the market. We have learned this in DS BPR as well. Number 9 and number 10 is the BPR document number and total number of pages in the BPR. The BPR is issued by Quality Assurance Documentation Cell, and there is a document format number for every document that you don't need to know. It is for the documentation department. That is all for the top section. You need to check this section every time you receive this BPR from QA documentation department or before executing any operations. The next section is who prepared this document. QA documentation person shall be the one who signs this row. This top row in green is signed at the time of issuance of BPR to you. As a manufacturing technician, once manufacturing team has manufactured the drug, they will sign in the second row, which is yellow. The third row in gray is for QC to check all testing-related data, and they will sign after their work and hand over the BPR back to you. The next row below, which is light blue colored, is for Quality Assurance Operations Department. This QA person will review BPR for any mistakes after manufacturing activities are done. This QA person reviews the BPR for any GDP or GMP errors. This is the QA person who also is present on the floor, and the same QA person gives the line clearance. Last but not least, once this BPR is checked by the quality assurance person, then this person will sign in the last row before sending the drug to the market. Since the drug is vetted properly now to be safe, after all required safety testing are completed, this BPR would go to the QA department again for reconciliation. Remember, this BPR was issued by the QA documentation department. Therefore, it shall go back for reconciliation and archiving. This is an FDA requirement. This is all the importance of the first page. A good technician will make sure the drug name and the product lot number and all related information on the first page is accurate and no signatures are missing. As usual, the very first part of a BPR is the BOM bill of material. To revise again, anything and everything needed to manufacture the DP will be mentioned in this section. As you can see, propranolol and citric acid are the DS and excipient respectively, circled in orange and blue, which will make the DP injections. Next is the red circle area where you see this sterile filter of 0.2 micron. This filter will be used to filter the drug to ensure no microorganism can pass through the filter. This filter will transfer the DP from formulation vessel to fill vessel. This fill vessel will be attached to the filling machine to fill the syringes. Moreover, below in yellow highlight, 1,240 syringes are mentioned. Targeted or theoretical yield is 1,200 syringes. Other 40 syringes will be taken for samples. There would be so many materials listed in BOM. This is just a representational BPR that covers the complete DP manufacturing process. Let's check your knowledge here right after you have gathered the materials from warehouse as per bill of material. What is next? Yes, the next step is sterilization of materials in autoclave. We have learned about autoclave in autoclave training already before coming to this class. And we understand in a nutshell that materials would be gathered in trolley under a certain pattern and loaded into the autoclave. Once sterilization is completed, the door of autoclave on the other side, which is cleaner, will open. You will take the autoclave printout and attach it to the BPR and autoclave log. But you must ensure that the autoclave cycle has passed. If it failed, inform supervisor. We have learned autoclave operation in the autoclave training, including the daily moisture test and vacuum leak test. That is mandatory every day before autoclave machine can be used for GMP operation. Now let's see what's on your screen and how BPR steps are instructing you to execute autoclave sterilization. Step 2.1 is instructing technicians to ensure that autoclave itself is cleaned and cleaning is recorded in the log. Technician will check and record yes for clean status and verifier will verify. Step 2.2 is instructing to ensure that moisture and vacuum leak test for the day has been performed and status shows okay. Moreover, the printout of this moisture and vacuum leak test is attached to the log and indicates a pass result. Step 2.3 and 2.4 instructions are directing to load both autoclave cycles as per defined procedure and press the required load number. Now, let me take you to the memory lane from autoclave operation training. If you remember that we have trained you on how autoclave trolley has multiple shelves. Each shelf has certain material spot and the material should be placed exactly at that spot allocation on the shelves. Each and every material will be placed at its designated spot in a way that the steam heat inside will circulate evenly to kill any microorganism present on the surface of the material. This is the same exact principle being applied here. Certain autoclave cycles are already programmed into the autoclave machine with a load setting number. That is the load number you need to press on the autoclave screen. Step 2.5 then directs you to review the autoclave printout to see if it has passed, sign, date, and add the lot number to both autoclave printout. Record each cycle's start time and end time. Verifier will verify with sign and date. That's all for autoclave and sterilization process. Moving forward, we will do drug formulation and then we will go through step-by-step instructions in BPR to fill our DP in syringes. And that's all for the DP process. Last but not least, if you remember from the previous autoclave training that once autoclave cycle is completed, the other door of the autoclave, which opens on more cleaner side, will open. Technicians on the other side of the autoclave will take sterilized materials to formulation and filling rooms accordingly. One more time, the line clearance term has arrived. Before beginning operations, the line clearance step is very critical We have also talked about line clearance many times from terminology class, day-to-day responsibilities of manufacturing technician, and DS, API manufacturing training as well. Now, this is the place in BPR where you have to take line clearance before you start performing any action. Quality assurance person will come to the manufacturing floor to ensure all materials are gathered in manufacturing area. All equipments are properly cleaned and status tags indicate that they are ready for use, as you can see on the screen in orange circle. After checking all is okay, QA signs off at line clearance space, as you can see it on your screen circled in green. Now let's move to the step number 3.1. How many of you remember glove box operation training? I am sure you all do. Concept of this glove box has been used here for DP formulation. Since we know that all aseptic and sterile operations must occur in grade A and B, this formulation is not grade A or B since there is no environmental monitoring required. This drug being produced will go directly into patient's blood as injections. Therefore, the air has to be controlled for absolute protection of the drug. We know how the glove box look like from the glove box training. At step 3.2, technicians are instructed to check the glove box is visually cleaned and log reflects that glove box has been set up and ready for operation. Technicians will record the status of glove box as ready in activity column. Verifier will verify the activity. Step 3.3 is instructing you to check the sterile gloves are appropriately attached to the glove box. Technicians will check if the glove attachment links to the glove box are secured tightly to prevent any leaks. Step 3.4 is instructing technicians to perform the glove box operation as per SOP 123. This SOP will provide step-by-step instructions on how to open DS, excipient, and mix them inside the glove box. Step 3.5 also talks about not to open any containers of DS or excipients until oxygen level reaches 0.3 or less. We have learned this concept in the glove box operations training. Just to repeat, at the oxygen level of 0.3 inside the glove box prevents anything inside from any microorganism contamination, since nothing can survive at this low level of oxygen. Even a tiniest bacteria or any living being that can be risk to the drug for patient safety. Moreover, if the chemicals being used are harmful, it is protecting technicians as you use the glove box. The glove box can also protect from any damage from spills and other accidents during the formulation. A company may have many reasons to do the glove box operations for certain activities. As long as you understand the operations, the concept behind the glove box and day-to-day operations of it, you are a good technician who can handle glove box operations regardless of the purpose it is being used for. Since you have all ingredients to be mixed within this glove box and the oxygen level is less than 0.3, you can start opening and mixing these chemicals together. Air inside the glove box is now completely safe. Step 3.6 is instructing to collect water for injection in formulation vessel, and water quantity is 5 liter. After adding 5 liter to the formulation vessel, you will record the liter quantity in next section. Verifier will verify. Step 3.7 is instructing to take the DS from the polybag. Step 3.8 is instructing to add DS into formulation vessel. Here you will record the weight of DS. Verifier will verify. Ensure that the item number for our DS matches. This is an extra step to confirm it with item number for assurance of accuracy in formulation process. Step 3.9. Add citric acid monohydrate powder, which is our excipient, into the formulation vessel. Remember, the sequence here, step 3.7, cannot be executed before step 3.6. Although you may have all the chemicals in glove box side by side, follow the step order. Step 3.10 is now instructing to stir the mixture in formulation vessel at 100 rounds per minute speed. You can control this speed from regulator attached to formulation vessel and make sure to mix it for at least 6 hours as defined in the step. Technician will also record, start, and end time of mixing so a reviewer can calculate and confirm the authenticity of mixing time at the time of review. This parameter is established for traceability and accuracy of this GMP operation. Step 3.11 directs you to take the sample from the sampling valve once 6 hours of required mixing is done. It is believed that after 6 hours of mixing, all attributes of this drug product DP will show in the test as achieved. We had done similar IPC sample test in DS training as well, where this sample was taken after the chemical reaction was done in bioreactor. These IPC tests are conducted usually right after the chemicals mix together to ensure the desired chemical product has been achieved. This step is also instructing technicians to check the clarity of the solution, that it should be clear and colorless. Also record the sample attributes in activity column, including sample time and volume in milliliters. On your screen, you see Module 4 of your BPR for DP process. Before we move further, let's recap again on what we have done and how much is left till the finish line. We gather all the materials from warehouse based on the list from bill of materials section. We cascaded materials into the clean room, then we sterilized the material using the autoclave. Sterilized material then was picked up and was transferred to formulation and filling rooms accordingly. In formulation room, you formulated the drug in glove box and sent IPC samples for analysis, which passed, and you attached the test results to BPR. Now, this module of aseptic filtration is an important one to understand and learn. Step 4.1 is instructing technicians to visually check if the fill vessel appears clean and log confirms that the fill vessel is ready to use or the clean status tag is affixed. Step 4.2 is to check all pressure gauges, nitrogen flow meter are within calibration. If you look at the yellow highlighted right in the middle, which says that DP is transferred from formulation vessel to fill vessel through the Sartoflor filter with a membrane of 0.2 micron. Let's understand the concept of this filtration process, how and why we are doing this step. Let's imagine you have your formulation vessel on your left and you have DP inside this vessel that you have manufactured, taking utmost precautions. Now, for an extra precautionary step, we are going to transfer this DP into a clean and sterilized fill vessel. And while making this transfer, we will also strain the DP with 0.2 micron filter to stop everything and anything that can damage the DP at this step. This fill vessel then will be hooked up to the filling machine where syringes will be filled and later stored for the next process of FDP. Further step 4.3 is directing technician to check the filter integrity. Let's understand how to check the filter integrity. Once you have transferred the DP from formulation vessel to fill vessel using the filter, which is supposed to block anything that is size 0.2 micron, usually the tiniest living being is 0.2 micron. Now you get the point? Anything that is size 0.2 micron or less will get strained. These filters come sterile from the manufacturer or supplier. You just use them, but how to ensure that the filter you used was not compromised, or in other words, if it had any leaks, the leak test is the best way to confirm and to ensure that our drug is safe up to this point. Filter integrity test machine is provided by the filter manufacturer, which applies air pressure to the filter and senses any abnormal flow of air to determine any rips inside of the filter. That is it. Now you know the reason of this filtration process and filter integrity and its importance for drug safety, as this is the last time to ensure the drug safety before it goes into syringes. The last note highlighted also mentions to notify supervisor if the filter integrity test fails. One more time, the line clearance term has arrived. Before beginning operations, the line clearance step is very critical. We have also talked about line clearance many times from terminology class day-to-day responsibilities of manufacturing technician and DS, API manufacturing training as well. Now, this is the place in BPR where you have to take line clearance before you start performing any action. Quality assurance person will come to the manufacturing floor to ensure all materials are gathered in manufacturing area. All equipments are properly cleaned and status tags indicate that they are ready for use, as you can see on the screen in orange circle. After checking all is okay, QA signs off at line clearance space as you can see it on your screen circled in green. The final step of DP process is to fill the drug product into smallest units defined. We have in this case 1,240 syringes to fill 1,200 syringes to send for labeling and packaging operation after this, which is the next stage of FDP. The filling of syringes require the environmental monitoring, and the drug that goes directly into your blood has to be manufactured under aseptic conditions, which in other words means grade A and grade B. We have learned this in environmental monitoring training. If you can see on your screen that QC person is responsible for environmental monitoring, including settle plates, air monitoring of viable and non-viable particles, both. The second note, highlighted in yellow, is instructing technicians to keep spraying the IPA on their hands every 15 minutes. This is a requirement during the critical operations in cleanroom. The practice is to kill any possible germs that may end up on your hands during activities. This is an extra precautionary measure to safeguard this critical operation. Step 5.1, 5.2, and 5.3 are for QC person's responsibility. It is not for manufacturing technician. Your job is to just ensure that when environmental monitoring is required as per BPR instructions, you must not start the operation until environmental monitoring has been started by the QC individual. At the second part of the screen, you see a table. This is entry and exit log to the filling room, which is inside this BPR to keep track of who entered the filling room and at what time, as there is an important GMP compliance related to it. Most cleanrooms with critical operations are air monitored. Therefore, only certain number of people can be in at the same time due to the relation of the ratio of the breathing and the size of room. In simple words, you will signs on the cleanroom doors about the maximum number of people allowed inside at the same time. This is to control the quality of the air, as this air is the same in which the drug is being formulated. This entry-exit log will be reviewed by the QA operation person to see if accidentally anyone entered and violated the maximum person allowed limit, which could be a deviation. Step 5.4 is instructing technicians to assemble the machine parts before filling and purge the nitrogen for 5 minutes. Let's understand what machine parts are being assembled. If you look at the picture on your screen, this is an automatic 5-head filling machine. In this machine, there are certain parts that get detached after every single use to get cleaned after each use and fill. These parts get cleaned and get put back into the autoclave for sterilization before these parts are brought back in for the next filling. And this is the time when these parts are being put back. After all the parts are connected together to the fill machine, this step is instructing you to purge nitrogen through the filling lines. Step 5.5 is instructing technician to connect fill vessel to filling machine. This is one of the critical step. You will be provided a special training before you are given this responsibility. Step 5.6 is instructing technicians to charge the rubber plugs for syringes into the filling machine hopper. Hopper is nothing but the entrance where you can drop all the rubber plugs, and machine will take these plugs one by one and put them on syringes after syringes are filled. Step 5.7 is instructing you to start the filling machine. Step 5.8 is a special note that all manufacturing technicians in this operation must place all 31 tubs of syringes to the conveyor belt one by one as the machine has started and conveyor belt is moving and taking these tubs under the filling needles. Step 5.9 mentions technician must record the start time of the first tub placed on conveyor belt. This way, after the end time, we will be able to calculate how long the filling machine took to fill 1,240 syringes. Step 5.10 is instructing to pull samples as per the sample plan, SOP number 331. Every company has certain plans to pull random samples from random locations. Here in this step, we will also record the total number of samples. Step 5.11. At this time, you are instructed to perform a visual inspection of all syringes in the tub to check any signs of leakages on the syringes or if any plugs are not sealed properly or any visual defects in the syringes. If you encounter any syringes for rejection, please record them at this step as all the calculations will be reconciled later. Step 5.12 talks about that technicians must record the end time. Step 5.13 then subtracts the start time from the end time and calculate the total time, and verifier shall verify the calculation for accuracy. Step 5.14. All 40 syringes that you have taken as part of random sampling, you must put the sample labels on each for what kind of test these syringes are aimed for. Such as, if you can see on your screen that you will be taking 20 samples for stability, 10 for microbial testing, and the rest of the 10 are for the assay confirmation. You have learned about these tests in terminology class. You don't really need to know the science behind these tests. Just need to know how to take affix labels on the right sample, making no mistakes, while at the same time creating traceability and accuracy for the drug and patient safety. Last but not least, the highlighted note at the bottom of the page. This note is instructing you to calculate the time between the start and the end. This time should not be more than 72 hours. What that means in layman's term, that means no matter how delayed the filling is, for any given reason, it should not pass beyond 72 hours.